to understand here. So it all starts with the disease condition. So what is disease? So disease is an abnormal condition. Yes, it is abnormal, but what is that which is really going abnormal here? So it is the protein which is behaving abnormal. So in the disease condition, there could be two types of abnormality with the protein activity you could observe. So either there could be increased activity of protein or decreased activity of protein. Now, yes, so how this could be happening, right? So there is over or underactivity of the protein. What could be the reason or how this could be happening? So if there is increased quantity of protein, so we could expect if there is over expression of the protein, then there could be overactivity. Or there could be the second reason, the quantity is same, but there could be increased inherent activity of the protein. How this could happen? So majorly because of the mutation effect. So because of modification in the protein, there could be overactivity of or underactivity of the protein. So since I am talking about protein, just to give the brief background here, so I'm very sure you are aware about all these things. Just I'm putting things together so that you could connect and you would get a better imagination, right? So that is the plan here. So since we are talking about protein, protein is polymer of amino acid residues. There are 20 different types of amino acid residues. Again, you could class them or you could group them together based on their nature. So there are hydrophobic amino acid residues like isoleucine, valine, right, leucine. There are aromatic amino acid residues like tyrosine, tryptophan. There are charged residues, right, positive charge, negative charge, polar, right. So now these amino acid residues polymerize. So the amino acid information is called as primary structure, right? So these would form a pattern of hydrogen bond. So that makes the secondary structure. So it could be alpha helix, beta pleated sheets, right? So these secondary structure unit form a globular shape of the protein, and this type of multiple tertiary structure could come together, form a quaternary structure. So if the protein loses this tertiary or quaternary structures, it loses activity. So what we call it as denaturation. Okay. Now protein could exist in um, different varieties. So you could class them as enzymes or the receptors. Okay, so what is the basic difference between these type of protein? So what we call as enzymes or the receptors. So enzymes are mainly involved in bringing some sort of catalysis. So digestive enzyme, you might have heard, right? So receptors are mainly involved in the signal transduction, right? So both are proteins. So both are made up of amino acid residues. So basically, if I call enzyme or receptor, you should not get confused because both behave exactly same way. So when we talk about proteins, it could be enzyme, it could be receptor, it could exist in active form or inactive form. Okay, so what is the difference? The observation here is observation here is when the protein is in active form. Okay, when I say protein, just imagine it could be enzyme or it could be a receptor. So if it is existing in the active form, if it is an enzyme, you would observe catalysis happening. Okay, there is a product formation happening there. If it is a receptor, it would propagate a positive downstream signaling. Whereas if it is in the inactive form, you wouldn't be able to observe any catalysis happening. Or in case of receptor, there is nothing like idle. It would be negative downstream signaling. Okay, opposite to the positive signaling, it would be propagating. This is the observation. But how this is happening? What is the reason protein behaves in different forms? So in one form, it is able to bring the catalysis, and in another form, we call it as inactive form, or where the catalysis doesn't happen, right? So the basic difference here is the shape of the protein. So here there is a graphics you could see. So in one shape, protein is able to bring the catalysis. We just call it as active form. In other form, it is not able to bring the catalysis. We call it as inactive form. So here is one example of kinase protein. And here is one receptor, GPCR receptor I have mentioned here. Okay, so what is it causing or what brings this differentiation? So in one form to other form, how it gets, how the shape changes. So mainly it could be because of substrate binding. So when I call substrate, so you could imagine there is a, if it is a kinase class of protein, you could imagine ATP is the substrate. So when ATP binds, it would induce this type of conformational change. So substrate could induce this type of conformational change, or it could be upstream proteins which are involved, which could push this type of conformational change. Okay, so let us simplify our imagination. Let us just focus on the substrate. Okay, so here you could see in the graphics, so there is a pink color molecule. You could see once this pink color molecule binds, it is inducing the conformational change in the protein. Now, substrate is very special. Why it is special? Because it has two unique property. The first property is affinity. The moment you put it with protein, it just goes and attaches itself to the protein. That is called as affinity. And the second thing is intrinsic activity. So when this substrate goes and binds, it activates the protein and you observe a physiological changes happening. Okay, catalysis would start, receptor positive downstream signaling would start, right? That type of thing, intrinsic activity. Okay, so normal or simple organic molecule will not have this type of property. Okay, so what is the driving force for the affinity? I mentioned the moment you put the substrate with the protein, it just goes and binds to the protein. What is the driving force there? The driving force is non-bonded interactions. Okay, so these are very weak type of interactions. So there is no permanent covalent bond happening. So little bit chemistry I'm talking here. So there is no covalent interaction happening here. Rather, it is weak type of interaction. So these could be hydrogen bond, hydrophobic interaction, electrostatic interactions, right? Dipole-dipole type of interaction. But the speciality is, even though their individual contribution is very weak, but you would see multiple these type of interactions. So these will have synergistic effect. So it is not the summation effect. So 1 + 1 will not be just 2. Rather, it would be synergistic. 1 + 1 could become 4, 5, 6. So even though their individual contribution is very less, so since multiple these type of interactions you would observe, they will have a very strong influence. So always remember, whenever you are analyzing interaction, you should be really sorry, I have a little bad voice today, so please bear with me for that. So whenever you are analyzing any interaction, you should be seeing all the type of interaction. Just don't think that only hydrogen bond is important or only electrostatic interaction is important. So there is a problem. So in the disease condition, the protein is either overactive or underactive. Let us imagine protein is overactive. It could be because of increased quantity of protein, or it could be because of mutation. There is.
Sir, if you want, you can have some water. No problems. Please.
Yeah, thank you.
Yeah.
I'm really sorry. So afternoon it started aggravating. Otherwise, it was all fine.
No problem, sir.
Yeah. So we were talking about disease condition. So there is either over or under activity of the protein. Let us imagine there is a overactivity. It could be increased quantity, or it could be because of increased because of mutation. There could be increased inherent catalytic activity. So if I have to treat the disease, I have to suppress the protein activity. So how could I do that? So if protein continuously changes the shape, then that would propagate positive signal, and that would further aggravate the disease condition. So if I prevent the shape change happening in the protein, then I would be able to control it, right? So how could I do that? So very simple mechanism you could imagine is the place where substrate goes and binds. In the same place, if I come up with a design of a molecule, an organic molecule, it could be what is organic molecule? The molecule which is made up of simple carbon, oxygen, nitrogen, sulfur, these type of atoms. Okay, so I could synthesize that it could be synthetic origin, which I have synthesized in the lab, or it could be a phytochemical origin, like Ayurvedic phytochemical compounds. Now, this molecule would go and occupy the same space or the place where the substrate is binding. Now there is a competition. So in the competition, now if the substrate I mentioned already, it is very unique molecule, right? So if it binds, so it has affinity and intrinsic activity. If substrate binds, it would trigger this shape change. But our molecule is not really a special molecule. Okay, it will have if it has to win the competition, because substrate is always there, ATP is always there in the body and it is there in abundance. Whatever medicine you take, it would be in very small concentration and very short duration you are taking, and in that concentration it should be able to exhibit its activity, right? Preferentially it should go and occupy the space. That means if in that competition, if our molecule has to win, then our molecule should be able to exhibit higher affinity, right? So that means if our designed molecule is able to offer more number of interaction, then in the competition it would win. So there is a competition here. So that's why it is called as competitive inhibitor. Okay, so this is this is one way of blocking the protein. So instead of that, so we could come up with a design of molecule where it could go and bind to some other place than the substrate binding site. We call it as allosteric site. So when our molecule goes and binds to the protein, you could see here initially the binding pocket was wide and open type, so that easily the substrate could bind to the protein. Whereas in presence of allosteric binder, you could see it has brought in a deformality. The shape has been altered for the protein. Now with this altered shape, the substrate is not able to bind. Again, it is like locked. So you have kept a stone near the hinge of the door, so door could not close properly, right? Open or close. So that's how ultimately, whether it is competitive inhibition or a allosteric inhibition, you are preventing the shape change happening in the protein. So once protein stops altering the shape, it would lose the activity. So this is very specifically applicable for enzyme class of protein. Okay, in case of receptor, the shape is very important. Okay, so if you want to have a positive effect, or if you want the protein to be continuously activated, you should lock the protein in the active shape of protein. If you want to inhibit, or you want to have an antagonist, then you should lock the protein in the inactive form of the protein. So in case of enzyme, shape doesn't matter whether it is active form or inactive form. You just block the shape change happening in the protein. But in case of receptor, the shape is very important. Okay, whatever type of effect you want to observe, you should lock the protein in that form. Okay. That is all about background. So let us try to understand about drug design. So is it just sufficient if molecule goes and binds to the protein? That's not that will not make a molecule drug. Cyanide is also a binder, but that is not a drug molecule, right? It has to be safe, right? So a lot of property it need to have. Of course, it need to have good potency. Addition to that, it need to have solubility, selectivity. It should be free of CYP inhibition. It should not have any hERG blockage. The molecule what we have designed should be synthetically feasible, right? So multiple properties need to be incorporated. So in the process, when we are going for drug design, we are trying to identify that one molecule having several properties in it, and this is what makes it complicated. You are not just seeing for one endpoint, rather you are looking for multiple endpoints in the same molecule. Right? That's why it is time consuming process. It is very expensive. So until you reach or you identify that molecule, you will have to keep trying. So every molecule you make, you synthesize it, you test it for several property. Now that is what increases the cost, right? That is what makes it time consuming. Right.
So, for user benefit, can you just tell what is hERG? Because many might not know about the delayed rectifier potassium channels. Can just give a brief on that? Yeah, little bit.
Okay, so I don't have much information about that. Okay, so now the hERG is mainly a potassium channel, so it's involved in cardiac rhythm, I believe, right? So
yeah, hERG is actually a potassium delayed rectifier channel that is actually involved in cardiac repolarization. So they call it as the human ether agogo related gene. So usually any kind of drug or something which you are synthesizing, if it has a HERG blockade, then it can have some repercussions on your cardiac pacemakers. So that's the reason we have to optimize whether it is going to block, how much is the blockade and everything. Just for users information, sir. Thank you.
Thank you. I could have a water and that
Yeah.
gave me some relaxation.
Yeah,
Thanks for
yeah.
that. Right. Okay. So now that's why it is really taking long time. So you have to optimize a molecule for several properties, right? So that's why there are several stages in drug discovery. Okay, so it all starts with target identification. So you identify a protein and you confirm knocking down that protein or inhibiting that protein results in actual improvement in the therapeutic activity, right? So it would benefit improvement. It would benefit in improving the health condition. Okay. The next stage is hit identification. So here you try to identify a chemical molecule which could at least bind to the protein. It would show at least some or minimal activity. Okay, the potency will be very less. In the next stage, you fine tune that molecule. You bring a lot of chemistry modification in the molecule so that the molecule's binding potency increases. So highly potent molecule you will have at the end of hit to lead stage. Molecule will have high potency, but it may not be safe. It might be having several side effects. So in the next stage, that is called as lead optimization, you try to remove all those bad properties. So how do you do that? So it would be generally because of some functional group, some chemical moieties which is present in the molecule, which would make it to bind to all these type of what I was listing, right? Solubility, CYP inhibition, HERG blockage. So these functional group could make the molecule to bind to those off-target proteins, which would lead to the side effect. So in the next stage, that is lead optimization stage, you try to remove that and fine-tune further the molecule so that without compromising the potency of molecule, you you make the molecule safe and selective. So during the same time, you would be checking molecules activity in preclinical stage. So that is in the isolated enzyme or in the cell lines, right? So or in the animal, right? In vitro, in vivo testing you would be doing. So once you get a decent, good quality result, so you would approach the authorities, okay, approving authorities. You would ask their permission for human trials. So in the human trials, there are three stages: phase 1, phase 2, phase 3. So every stage again authorities would monitor. So if molecule is safe and is able to exhibit its activity at the end of phase 3, you would get a approval. Then you could go for marketing of molecule. Still, phase 4 trials would be continuing. So overall, it would take at least 10 to 13 years and more than $2 billion involved, right? So is there a way you could cut short this? So uh, about 5,000 to 6,000 molecules are synthesized, tested. So can you reduce that number of molecules required to be synthesized, or can you cut down the time so that the expense would reduce? And that is what the computational tools could do it. So even before you synthesize the molecule, you would be able to predict the activity. So what would be the potency of molecule? Would it have side effect? Can it touch other proteins, right? Selectivity. What would be the solubility of molecule? Can it have HERG activity? Can it have CYP inhibition activity, cytochrome P450 activity, right? So all those things, even before you synthesize, even before you do the testing, you would be able to predict in the computer. So only the best molecule predicted, very safe, very good, very potent. Only those molecule now you could take it to lab and you could test it. So that's how you could reduce a lot of time, because doing testing, the things in computer doesn't really take much time. So you could reduce drastically the time. There are two approaches. So when we are talking about computational drug discovery, there are two approaches. The first approach is called as structure-based drug discovery, and the second approach is called as ligand-based drug discovery approach. In structure-based drug discovery, we literally use the information of the protein. Okay, so where is the binding site? So protein is made up of amino acid residue. mentioned, right? So now there would be different types of amino acid residue. There could be polar residue, there could be charged residue. So if our molecule has to bind, it has to complement the amino acid residues present in the binding site, right? So you analyze the binding site, and based on that, complementing the binding site of the protein, you come up with the design of molecule. So that is called as structure-based drug discovery. Okay. In ligand-based drug discovery, so in many of the cases, getting protein structure is very difficult. It's a polymer, like plastic. So you have polythene cover. So it is very difficult to crystallize polythene, right? Same way, crystallizing protein is very difficult. You see, table salt and all, those are very easy to crystallize. So if you make a heated solution, super saturated solution, and if you cool it down, you get beautiful needles. For small organic molecule, that happens. But for polymer, crystallizing protein or polymer is very tough. So very few crystal structures are available for the target. So for all the protein targets, getting or going with structure-based drug discovery may not be possible. So in that scenario, we go with ligand-based drug discovery method. We use ligands information to come up with a hypothesis. What should be there in the molecule to exhibit better potency, or what are the problematic parts present in the ligand which you could remove to make it better molecule. Okay, so here there are approaches like pharmacophore screening, fingerprint screening, nowadays artificial intelligence, machine learning, right? So neural network, these type of technology uses ligand-based drug discovery method. So in my presentation, I would be majorly talking on structure-based method. Okay, but I would also cover few of machine learning or artificial intelligence method also as the application. Okay, let us try to understand structure-based drug discovery method. So very simple representation is lock and key. So when the substrate going and the way you represent substrate going and binding to the protein is like lock and key. So what I mean to highlight here is, even though it is very simple representation, it is very meaningful and very detailed representation you could imagine here. So the key perfectly complements the lock. Then only it would be able to open the lock, right? So there are several teeths in the key. One of the tooth if breaks, lock will not open. Same thing applies to the ligand also. So if a ligand has to tightly bind to the protein, it should perfectly complement. So I'll give you an example. Let's imagine this green color thing is protein, and the substrate, let us imagine it is round. So you could see here, if it is round, still it would have occupied the space, but there would have been gaps here, right? So the gap would be filled with water. Inside body, everywhere water is there. There is no vacuum, there is no air bubble. Everywhere water is there. And water is the greatest flusher. It could wash out everything. It can corrode metal, it could corrode stones, right? So anything it could wash out. And this is a small substrate, small organic molecule, it would wash out everything. Okay, that's why if it is perfectly complementing, so if you put a groove here, then it would perfectly occupy the space. There won't be space for water to wash out it, right? So at least for a longer time, it would be existing with the protein. So it would be able to exhibit better activity there. Okay, so how do you find out how molecule binds to the protein? So I was showing you complementarity, all those things, right? So we need to figure out, is there a way with which you could find out how a molecule binds to the protein? So here is a graphic picture. You could see this is a protein. So you could see surface. So I'll just show you again. Oops. So oops, right. Yeah, so this is the protein. You could see here secondary structure of protein, helix, beta pleated sheet, and all these are all different amino acid residues represented. It's a tertiary structure of protein, right? So in this protein, now you could see protein is shown as shape, right? So with the surface, we have shown something like you are covering protein with the blanket, right? So in the protein, you would be able to see some shallow pockets or deep pockets you would be able to see. Most of the time, organic molecule would bind to deep pockets. Okay. Now, depending on the pocket, the way the ligand occupies might change, right? That is what we are trying to identify. So with molecular docking program, so you might have heard molecular docking several times, right? So with this program, you are figuring out what is the way the ligand is occupying or binding mode of the ligand. And the second thing is, what is the strength, binding affinity, with what potency, right? It is binding. Okay. So what was the driving force for binding affinity? It's non-bonded interaction. The molecule which exhibits higher number of interaction, you could see here number, higher negative number indicates stronger binding, right? So the molecule in drug discovery process, majorly for potency optimization, we are looking for a molecule which could bind efficiently with the protein with multiple interactions so that it will have higher binding affinity. Let us little bit try to understand about the binding mode. How does software or how a molecule would prefer to bind to the protein? Okay, this is something very simple, very layman's language. Let me try to put this. Let's imagine you have different types of chairs. Okay, there is one tripod stool, there is normal uh, chair, plastic chair, there is a better chair, sofa like chair, okay, wooden chair, and imagine there is a very good sofa or bean bag. The way you sit on all these chair would be different, right? What is your focus while sitting on the chair? You see for maximum comfort. So in the tripod stool, you have to sit erect because there is no back support there. But when you are sitting on a sofa set, you take a posture which will support whole of your body, right? It will have hand support, it will have back support, it will have neck support, you will have head support, right? The way you sit on sofa, if the same posture, if you take on tripod stool, you would fall down, right? So basically you are seeing whether the posture what you have taken as a complex. Now imagine chair as a protein and you as a ligand. So when you sit on the chair, both chair and you should be comfortable. You also should not fall and it should be comfortable for you. It is physics driven, energy driven process. Whatever is relaxing way, that is the posture you are taking. And the same thing happens with the ligand even. Okay, it tries to occupy in most relaxing way. Okay, but is it lowest relaxing way or lowest energy? No, it is not. Right. So for us, lowest energy posture is lying down, sleeping posture. But we don't sleep on the chair. I mean, we don't take that lying down posture. You can't work like that, right? So it's a different posture. It is something. So if you stand upright, it is a high energy posture, but it's not high energy posture we take on the chair. Rather, it is something in between. And the same thing happens here also. Okay. So how does software figure out what is the best posture? So you are changing your shape, right? So when you're sitting on a tripod stool, your shape is different. And when you're sitting on a bean bag or a sofa, your shape is different. The same thing software has to do. It has to explore different shapes for the ligand. How do you do that? So in any docking program, okay, so what it has is two component. The first thing is generating different conformations or generating different shape for the ligand. So how do you do that? If there is single bond, you could rotate, you could change the dihedral angle and you could generate different shapes, right? So now let us imagine there is a single bond here and there is a single bond here. There is a single bond here. So if I give 1 degree increment, so total rotation would cause 360 degrees. So 1 degree if I give, so here 360 shapes I could generate for this bond. There is one more single bond here. So I could generate 360 into 360, all permutation combination I have to try out, right? So 360 to the power 2. There is one more single bond here, 360 to the power 3. Imagine the number of samples generated now for exploration. Once the search algorithm generates all those samples, now it puts one by one into the pocket. Okay, and now the second component comes into picture, that is scoring function. This will calculate what is the binding affinity, what is the strength of interaction. So we already discussed. So interaction affinity is because of non-bonded interaction. So it calculates energy component coming from each type of interaction. So energy of van der Waals interaction, electrostatic interaction, hydrogen bond interaction, hydrophobic interaction. So all those type of interaction component, it would consider, and from there it would calculate the binding affinity. Now it's not summation, just to make you understand I have put here plus, but it is some statistical equation, regression equation we have generated. Okay. Now before we go into the docking, we need to process the molecule. You might have seen in many textbooks, or whenever we sketch the molecule, we show molecule like this in 2D. So how each and every atoms are connected, that is what we are showing. But in reality, molecule doesn't exist in 2D. It exists in 3D. So geometry optimization is very important aspect, right? Not only just the geometry optimization, there are several other factors. There would be chirality definition. There would be tautomeric forms possible. There might be different ionization state possible, right? So all those things has to be taken care. In Schrodinger, we have a tool called as LickPrep, which easily handles all those things. In one click button, it will take care of all those things. Similarly, you will have to also process the protein structure. Structure based approach we are trying, right? So first of all, where do we get the structure of the protein? There is a database called as Protein Data Bank for free of cost. Okay, for free of cost, you could download all the structures available in that database. Okay, in the globe, whoever solves the protein structure, they deposit there. Okay, there are different techniques available now. X-ray crystallography is there. Electron microscopy technique is there. NMR technique is there, right? So preferentially we take X-ray structure. There would be a lot of lacunae associated with the structure. If you are working with X-ray structure, there won't be hydrogen. If there is no hydrogen, there is you won't be knowing what would be the ionization state of the residue, amino acid residues, right? Aspartic acid. So in one environment, like aspartic acid could in now it's the microenvironment which is important, right? So in one environment, it may exist as neutral form, COOH. The terminal residue atoms could be in the neutral form. The same aspartic acid in the different location, when environment changes, it could exist as ionized form. Similarly, histidine, there are 5 different tautomeric forms possible. So all those things has to be properly understood by the software. So for processing the protein in Schrodinger, we have a tool called as Protein Preparation Wizard. In one click button, okay, in one click button, several proteins simultaneously you would be able to process, you would be able to prepare. Finally, it would give a complete structure, whatever missing residues, if there is side chains missing, everything it would automatically recognize. It would make it complete structure, minimized structure, it would give us a output. So I have now prepared ligand. So I have to go for experiment, right? So I need to prepare my all starting material. So my protein is ready, my ligand is ready, but I need to know where is the ligand binding, right? So I need to know where is the binding site. So if I take a protein which is already bound with the inhibitor structure, which is a complex structure, then I would know, I will have experimental evidence where is the binding site. Suppose we don't have that information. So experimental evidence we don't have. In that case, there is a software which would analyze. So I showed you in the video, right? So generally the ligand binding pocket is a deep buried pocket. Okay, sometimes it would be shallow binding pocket also. Generally protein-protein interface is shallow binding pocket. Small molecule or substrate binding site would be generally deeply buried. So these tool, so in Schrodinger, we have a tool called as site map. It analyzes the protein. And you might have observed when I showed you the graphics, the protein surface is not even, it's not flat. It is uneven. So it has deep pockets kind of things, right? So it would analyze all the pockets present in the protein and it would recognize which pocket could be a ligand binding pocket. So first thing it would check which pocket could be a meaningful ligand binding site. And the second thing what it would check is which pocket, if you target, you can come up with a druggable molecule, right? What is meant by druggability? A molecule will become drug if it has suitable ADME profile, absorption, distribution, metabolism, and excretion, right? So the property I was listing, if molecule satisfies all those properties which I showed you earlier in the slide, that means we are solving ADME issue, right? So by analyzing the environment in the pocket, it would be able to check whether the pocket is suitable or druggable pocket is it, is it a druggable pocket? That is what it would check. In fact, it rank orders. It would suggest you give first preference to this pocket, second preference to this pocket, like that. So once I have complete information for docking, the proprietary product what we have for molecular docking in Schrodinger is called as glide. The unique advantage here is you have a choice between speed and accuracy. So what is meant by choice between speed and accuracy? You have a huge database, then you have a fast method. Okay, I have millions of molecule or lakhs together molecule. Then I need a fast method. Then in that case, I can go with high throughput virtual screening method. Just 1 second it would take to complete the docking. Okay. Next precision level is standard precision, about 10 to 20 seconds it would take. And the most accurate method is called as extra precision mode. 100 seconds it would take, 1 to 2 minutes it would take. So what I could do is I have a huge database, 10 lakh molecule I have, which I would put for docking. So I would go with fast method. Fast method would be less accurate method. Okay, HTVS I would take up. From this, whatever results I got, best 1% or 5% of the result, I will take it. I will put it for standard precision. Now I reduce the number of molecule, but I increase the accuracy level. Further, I have the most accurate method, that is extra precision mode. So whatever results I got from standard precision, I take 1 or 10%, 1% or 5% of the top results. I would again confirm with extra precision mode. So whatever molecule I am considering, it's a smart method, fast method I'm using. Okay, quickly I'm scanning everything. Whatever molecule I finally picked, at least 3 times I confirmed. So that is the advantage. Here is the accuracy level. So compared to there are n number of docking programs in the market. So if you compare to any other software, the performance with Schrodinger Glide is much superior. The enrichment, the ability to identify a true hit from the bulk is called as enrichment. That is very high. Not only that, even the post reproducibility with the experimental experimental quality result is also very high with Glide. Let us try to understand some of the case studies. Okay, so if we have to use computer aided tools or computer driven drug discovery tool, then that means the technology should be really superior. So with the technology, whatever property prediction you make should be really very accurate. So ideally, whatever tools I am using, like for example, all these properties like potency, selectivity, solubility, permeability, all these property I should be able to very accurately predict. But are we there? No, not yet. Okay, but at least few of the property we are able to very accurately predict, like potency, selectivity, solubility, to some extent clearance, to a greater extent permeability. In Schrodinger we have very good tools. Very difficult to predict drug-drug interaction, synthesizability again complex, toxicity, multiple things comes into picture, right? Probably today we are not there. Maybe in next 5 years, maybe or next 10 years, we would be able to really predict all these things. Then whatever you design in computer, you just synthesize, it will be a perfect molecule and it could reach to clinical trial, right? But at least at this stage, few properties we are very accurately able to predict, like potency, selectivity, solubility, and with that we are able to bring the differentiation. Okay, so what is our approach? How we are doing differently than the normal or traditional approach? So in the traditional approach I showed you earlier, there are several stages. It takes really long time. Very few molecules are explored, actually synthesized. About 5,000 to 6,000 molecules are synthesized, and still there is high attrition rate. Molecule fail in clinical trial, phase 1, phase 2, phase 3 trial, it fails, right? So how we are doing in Schrodinger? We actually scan billions of molecule, crores of molecule we scan in computers. But when we go for synthesis of molecule, it is very less. So here in this slide I have shown 1,000, but really we don't go for 1,000. It's very small number, 300, 400 molecule, that's all. And with that, we are able to cut short the time. We are very strict in applying filtration criteria. So that's the reason the attrition rate is low. Okay, so let us, what happens if you compromise? So here I was talking about compromise, right? So what happens if you compromise on certain property? So our approach mainly is fail fast. So if there is a problem in the molecule, you identify it in the earlier stage itself so that you don't spend more money. So if you compromise on potency, then dosing would be issue. If selectivity is issue, then side effect would be a problem. If you compromise on some functional group, stability would be issue. If you compromise on solubility, formulation would be issue. And if you compromise on the earlier stage, later stage it would become very heavy, right? The cost would increase. If you fail the molecule in the preclinical, if you drop the molecule in the preclinical stage itself, then clinical trial need not be, you would save that money, right? That's the strategy we are trying here. Okay. So in Schrodinger, we explore large number of molecules, billions of molecules. So if you explore larger pool of sample, getting successful molecule or identifying a better molecule or getting success increases. Obvious, it is very simple to understand, right? So there are two ways with which we try to explore large library. The first thing is already existing database are there. There are lot of chemical companies are there who have a large pool. So ready to synthesize molecule or synthetically or ready to purchase type of molecules are there. So those type of molecules we could explore, or we have a lot of generative models. Okay, so there is artificial intelligence methods, reaction based enumeration methods. So we have tools with which you could generate molecules in the computer. That is one way. Second thing is we are very strict on applying filters. So first thing, you are exploring large chemical database. It could be purchasable database, or you generate de novo new molecule in the computer, depending on different methodologies. Okay. So once you generate those large pool, then you very strictly you apply filtration criteria. It could be very simple like molecular properties, or we have capability to predict physics based methods like binding affinity, free energy, perturbation, like type of tool, or we also have capability to generate machine learning model through which we could apply, or we could predict the properties and we could apply those type of filters. Okay. I was talking about docking. I said we have 3 different modes and that is the advantage. But nowadays you have crores of molecule, 100 crore molecule database you'll have. So with that, even if you go with HTVS method, you won't be able to complete the virtual screening in given time. If virtual screening itself takes like years together, 12 months, 18 months, 24 months, it's of no use, right? So we need to have a technology which could further speed up the process, right? For that, we came up with the idea of AL Glide. So here we integrated machine learning model in the molecular docking. So how exactly we are doing? It is something like iterative supervised learning. So something what is used in speech recognition or information extraction, right? Those type of things. So what exactly we are doing? Let's imagine I have 100 crore molecule collection. So if I go for 100 crore molecules docking, you just imagine even if I go with 1 second per one ligand, it would take multiple years, 4 or 5 years to finish the docking. So that's of no use, right? So what we will do? So from this larger pool, very small subset we will take. Let's imagine 50,000 molecule, 50,000 or 1 lakh molecule. Okay, first we'll go for docking. Now, since number of molecule is less now, only 50,000 molecule or 1 lakh molecule, I would directly go with intermediate standard or standard precision method. So I would dock those molecules. So now I can generate machine learning model between the structure of molecule to docking score. So once this model is generated, we will apply this model on whole of the database. Now, while applying this model, it would check for uncertainty. That means it would check what is the confidence level, whether the generated model is really able to correctly predict the activity. That is what it would check. So if the uncertainty is really low, very bad, that means the generated model is not good. Again, it would go back one more 50,000 molecule. It would take up. It would go for docking. So like this, the process would be repeated. The cycle would be repeated several times till you get a very good quality model. Once you have a good quality model, then the top whatever predicted molecule, top 1% or 2% of the molecule, it would fetch, it would actually run the full-fledged docking, and then it would give you the result. So you could see here the speed. with which we could complete the process. So let's imagine I have 120 crore molecule in my hand, and if I have so something like 25 CPU machine, I would be able to finish the calculation just in 22 seconds, 22 days. So in our in-house drug discovery projects, we have really huge number of cluster. We have huge infrastructure. So we use something like 500 or 1000 CPUs, and with that we are able to finish the virtual screening in just like 2 days, 2.5 days. So that type of speed you would be getting. So generating machine learning model will not take much time. So that is the advantage we are incorporating here. It's a smart approach. Did you really dock all the molecule? No, hardly few lakhs molecule only you are docking. But did you omit out any molecule? No, you exploited or you explored every molecule, and then the best molecule you fetched and confirmed it. That is the advantage here. So molecular docking could be used for initial virtual screening. So identifying, let's imagine you have 10 lakh molecule. From 10 lakh molecule, you want to pick some 1000 molecule or something like that. That would be useful. But from 1000 molecule, I want to pick 10 molecule which I want to consider for synthesis. Then docking will not help you. The docking scoring ranking methodology is not so accurate. Okay, so for that we have a technology called as free energy perturbation. Very accurate method here. Number to number you can match. So computational prediction could be matched with the experimental value. So enzyme wet lab enzyme inhibition study, if you are carrying out, the error margin is within 0.6 to 1 kilocalorie. That means it is equivalent. So whenever you run the assay, you would put duplicate or triplicates, right? So in that itself, you would see more than 1 kilocalorie error margin, and the computational method is within that. So how exactly we are working? So in the docking, we go in this direction, 1 and 2 direction. That means you take separated protein and ligand protein, you put them together. How much energy gain is happening when it forms complex, you are checking. But the system is in equilibrium. So we go across A and B. Okay. So in reality, this is not possible, right? So in computer, we could do it. So in the complex form, you modify one molecule to other molecule. Okay, you couple it with solvated system, you run MD simulation with that. So all the conformational flexibility, shape change happening in the protein, ligand, solvation effect, all these things you are considering. And with that, the ligand change, how it is contributing in the interaction, binding affinity, all those things you are checking. So slowly during the simulation from one molecule to other molecule, you are changing. So this is called as alchemical intermediate. Here you could see. So initially it is hydrogen. Slowly during simulation, you change the molecule to chlorine here, right? So with that, binding affinity increases. So this is one example with just one atom. So you could change whole of the core up to 35 to 40% of the molecule. You would be able to change it. Now this is what is changing the drug discovery scenario. I'll show you quickly show you a small example. So this was Roche paper. This is an old paper, but it was an interesting paper published in 2017. So I just wanted to take as an example. It was actually kind of competition. So 4 groups were working. So one group was using free energy perturbation for shortlisting the molecule. So one group did not use any of the computational tool, pure medicinal chemistry based experience based approach they were using. Other group was using classical molecular modeling tool like docking, pharmacophore, those type of things. And there was one group who was using internally developed Schrodinger's own product. See, those big pharma companies can have those type of things also, right? So all these 4 groups were asked to shortlist 10 molecules. So they were given with about 3,325 molecules with this type of scaffold with modifying here. Okay, so chemistry modification was brought in here. So 10 molecules. So the cutoff value expected is 200 nanomole. So enzyme inhibition should be less than 200 nanomole. So out of 10 molecule, who would be successful in identifying most number of molecule? That is what was the competition. So if you see here, Schrodinger, based on Schrodinger FEP, we had 80% success. The rest all group had only one molecule. That means 10% success. So these were the molecules. So this is the scaffold. These are the different substitution. You could see actual activity was 25 nanomole, computer prediction was 24. So here 27, 15. Of course, few molecules we went wrong. So 20% wrong we went. So when this type of thing happens, we We go back, we check what is going wrong with our prediction, right? So we refine further, improve the technology. That's how the accuracy is increasing. So Schrodinger, I'll quickly go through. I think I have another 5 minutes. Okay, I'll quickly show you a very interesting example. So Schrodinger not only does software selling, we also have our own internal drug discovery program. We also do joint development. So almost 35 to 37 drug discovery programs we have. I won't be able to share our collaborative program. I'm really sorry. IP issues are restricting us. We have our own drug discovery programs, about 6-7 programs we have. I would try to show you few of the things. Probably one case with limited time, one case I will try to cover. Okay, so this was one very interesting news news article published by uh, 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 chief computational chemist at Bayer. So they implemented our technology because they named it as Nova design. So automatically generating large library, then applying screening methodology, right? So all those machine learning methodology, free energy perturbation, docking. So all those things, they have automated technology we have provided them. So he published, he said that with this type of technology implementation, they could reduce the drug discovery cost by 7 times in multiple projects, and they could reduce the time by at least 3 times. So which was earlier taking like preclinical stage, something like 18 to 20, 12 to 18 months. Now they are able to finish just in 4 to 5 months, right? I wish I had those type of more example, but I'm sorry. But at least our internal things I will try to show. So what is the approach here? So how do we actually go about? So as I mentioned earlier, we explore large number of database. So either purchasable database, or we have a platform called as Auto Designer, which automatically comes up with the design of molecule. More than billion molecule it could generate. Okay, so once the billions of molecules are generated, we very strictly apply molecular filters. So it could be as simple as molecular properties. It could be log p value, polar surface area, right? So those type of things, Lipinski rule of I. It could be as simple as that, or it could be very complex multi-parameter optimization. So you apply the filter. Still, you will have millions of molecules. Now, all these millions of molecules, I cannot run free energy perturbation because it is time consuming. It's a molecular dynamics based calculation. Okay, it's very accurate, but accurate methods would be very slow. So what we are doing here is we randomly pick very small number of molecule. Let's imagine 1000 or 2000 molecule. You run a single edge, a simplified version of free energy perturbation, or single edge FEP. Okay, it won't be very accurate, but it is good enough. Okay, so with that you generate a machine learning model. So generated machine learning model you apply on whole of the database. Now you pick best predicted molecule. Now you run the full-fledged free energy perturbation. So from large number of pool of molecule, now you actually shortlist very small number of molecule. So from here you could see 5000 molecule to 10 molecule I'm picking, and in 10 molecule I'm sure that at least 80% success rate we will have. I'll show you an example. So this is one of the program which is in the advanced stage, V1. It's a kinase. So in the kinase program, the major issue is getting selectivity. So here we used computational approach to bring the selectivity. Okay, I won't go into the pharmacology of V1. So let me put you the story here. So the major problem here is again getting off-target effects. Okay, if see in the disease condition, one particular protein is problematic, overexpressed or overactive, but all other proteins normal activity is required. Now all kinases are modulated through ATP. So you could expect now if 500 different kinases are there, same ATP could bind to all those 500 molecules. That means you could expect binding site would be exactly same or almost same, right? So that means if I go with competitive inhibition, so I hope you are able to connect my earlier slide. So now if I come up with the competitive inhibitor molecule, now there is a high chance that it could bind to other 500 kinase also because binding site is almost same, right? So now bringing selectivity is the big challenge. So here again we were working with V1 where we wanted to bring the selectivity. So our competitor molecule from the publication, we could see that they had really selective molecule. When we started working on this program, So you prioritize what is your primary target and what is your primary off target. So here it was PLK1, which was a primary off target. So we got really fantastic selectivity profile. 1,000 times it was selective. So we thought we really reached to our desired level of selectivity. But when we did kinase panel assay, so every line here indicates different kinase. So red color dot indicates problem. That means they are touching all those kinase. So here you see so many red dots, and larger means it is non-selectively, it is going and binding and very potently inhibiting all the kinase. So you would expect really very ugly side effect here. So this molecule was really bad. So now the next challenge came into picture. So what do? We drop or we bring the selectivity? We took it as a challenge. We used computational tools here to bring the selectivity. Again, what we did, we enumerated huge database, okay, billions of molecules. So here, virtual screening, we went handwritten. So we have chemists also in our group. So they also sketched a few molecule, purchasable molecule, everything we explored. So the best molecules, then after applying all the filters, we went with FEP, free energy perturbation, to check target affinity and off target affinity. Then to bring the selectivity, we went through FEP kinase panel assay, and from there we selected the molecule. So here is, in short, I would like to tell you, we started with 445 million molecules, 4 crore molecules we started. So after applying all the filtration criteria, few million molecules were filtered. So about 6,700 molecule we considered for free energy perturbation, which exhibited high potency to the V1, okay, which were highly selective. Now we wanted to check whether these molecules are not touching any other kinase. Okay, so for selectivity, we shortlisted based on this. Now you could imagine each and every kinase we should take, and we should run the FEP. So you could imagine 500 kinase are there. If it is 1,500 molecule, you could imagine the extent of calculations we had. So about 1,500 molecule we could identify through FEP, which were highly selective, and from 1,500 molecule, only 42 molecules we shortlisted for the synthesis, and out of this 42 molecule, 22 molecule reached the desired objective. They were highly selective and they had very good kinase profiling. How exactly we did? Is it literally possible to scan all the 500 kinase? No, we used a smart approach. Okay, we analyzed all the kinases. So what is the modification happening in the particular place that we scanned, and with those modification we ran the FEP. So protein FEP we call it as. So with that we were able to calculate the binding affinity, whether the molecule is showing high selectivity or whether it is able to bind to all the different modified protein. Also, is it able to bind? That is what we were able to check. So we generated such about 20 to 25 models, and with that scanning, we could really identify highly potent molecules. So initially, the first molecule was not really great, but finally we had a molecule which were highly selective, more than 5,000 times selective to any other kinase. So here are the 3 molecules. So compared to AstraZeneca molecule and Genentech molecule, you could see the profile is much better, and these are in the advanced stage. This doesn't have CYP inhibition, highly selective over PLK1, right? So very good profile it had. So there were other projects, because of time restriction, I could not go through other stories. So you could see here, these are the number of molecules actually explored in computer. So every case it was more than 1 billion molecule. Okay, and what is the number of molecules synthesized? Hardly 200 molecule, 300 molecule, and with that we were able to identify clinical candidate. Okay, so summary of example. So I mentioned we have about 35 drug discovery project, and we started probably 3-4 years back only, and this is the status. We already have 2 FDA approved molecules. About 3 molecules are in phase 2 trial. About 7 molecules have started with phase 1 trial, and IND enabling investigational new drug application enabling studies are going for at least 5 projects, and we have about 10 potential best in class and about 5 are first in class type of molecules. So these are the references. So in my presentation, I just covered mostly into small molecule package. We also have material science suite. So formulation related things, excipient selection, why nanoparticle will form, or optoelectronic design, right? Samsung is our customer. Okay, they used our tool for designing their thinnest uh, uh, phone screen. Okay, we also have biologic suite, so antibody designing, vaccine designing, protein engineering tools, all those things we have. So with that, I would conclude. Really sorry I extended by 10 minutes. So if there is any question, I would be happy to take it.
Thank you very much, sir. I think this is one of the most complex topics in the life science domain, and I'm surprised to see how come you love chemistry so much. So it requires a lot of talent to do that, and a wonderful lecture, sir. You cannot get better than this.
Thank
Because only few people across the country, I'm sure, they have a little bit of knowledge about this background process that runs in the drug discovery.
Yes.
Everybody thinks drug discovery is just clinical trials, pull some money, we can do it. No, it's not that easy. There's a lot of work that goes, and now
Yes.
with the evolving technology,
Yes.
and I think rightly said, Schrodinger is also producing their own molecules and have got drug approvals. I
Yes.
think there can be a lot of avenues in analyzing a drug, whether it can cause an adverse event or
Yes.
not.
Yes.
With respect to your computational designing,
Yes.
you can even take up pharmacovigilance activities.
Yes.
So,
Yes.
yeah.
We haven't put our hand into that now yet.
I think then we should collaborate. So let's talk later about that, sir. No issues. So thank you very much. So let's have some couple of questions from the audience if they have anything. It's open to questions, audience. You can ask a few questions. So I have a question.
Yeah, please.
It's from a commercial point of view. So let's assume that I'm a researcher. I have done some quality preclinical studies with respect to in vitro, in vivo. I have tested few molecules. Let's assume that 10 molecules I have tested. And with respect to efficacy and toxicity and other things, I have tested completely. I feel my molecule out of 10, 3 had given me good results in the rank order. So now I want to check among a pool of similar molecules which you have in the library. So I want to run a battery of tests using the computational drug design. So is it commercially viable for an individual researcher to do this, or it's only for corporate companies, pharma companies?
There are many startup, single man startup companies who are coming up, and their process is totally driven based on computational tools. So we ourselves are running. So drug discovery team is a separate team. We have a firewall with them. So they, like, when I'm from technical support team, so we interact with customers and all. We provide the support. So because of conflict of interest, that group is totally separate. We don't interact with them. But we also run some service projects. So we get such customers where single man company who gives us the project, and he takes it to for all finalization, shortlisting of molecule. They'll use computational tools, and then they go for CROs to synthesize the molecule, get the molecule, sell it off, make money.
Yeah.
Right.
No,
there are several case
when you were
studies.
talking about, I just got this idea because you said Schrodinger itself is into that process.
Yes.
Then immediately my mind struck, why not I do it?
Yes.
As a businessman.
Yes.
So I just wanted to, because there might be other people who might have some good research which they have done, but they might not be able to take it to the next level because they might not know how viable it can be in a clinical trial. Even though you have a real world evidence data which you do, the companies do, I still believe that computational drug design influences your trial enrichment for sure.
Yes.
So, one more question I have, sir. So how is AI evolving in computational drug design? Is it like accurate, or is it like trying to become accurate?
Okay. See, any machine learning method depends on what the data you have. So how
Correct.
you train your model, depending on that, the quality of result would change.
Correct.
And in drug discovery, you really have very limited number of data set. So
Correct.
let's imagine binding affinity you are trying to predict. So how many in the globe would be working on one particular target, and how many of them would be publishing, or how many data points you would have? So hardly you might be having some 1,000, 2,000 data points, and that wouldn't cover all the diversity, right? So you might be changing chemistry in the molecule, so property would change. So that training would not be sufficient. So that is the major limitation with artificial intelligence. So you need to know what is the advantage and disadvantage of the technique, and based on that you should carefully use. So you can't say, my belief is, just based on artificial intelligence you can't come up with a molecule. So you will have to use different technology based on or depending on the different stages. You should choose different technology. Like I was showing you. So in the initial virtual screening phase, we use machine learning model. So it was docking based model. So accuracy is not again perfect. So but from 100 crore molecule, if I have to pick 1 lakh molecule, that works for me. But from that 1 lakh molecule, next I have to pick 100 molecule for which I'm going to synthesize. Now if I use those type of machine learning model, it won't be useful. Then I have to go for free energy perturbation like technology. So
But
there I would be able to. Yeah.
But you also mentioned that, for example, there are some databases with respect to chemicals. Some
Yes.
companies do have, but internally Schrodinger is
But
also
property will not.
generating different probabilities and different possibilities of different chemicals. So I saw in the slide 1 billion was in the database, but Schrodinger had 10 billion.
Yeah,
So
that is
using the de novo synthesis of chemicals, can machine learning be trained?
synthesis of molecule or design of molecule you could make, but you will not have property of those molecule, right? So if you ask me what would be the liver toxicity of that molecule, or what would be cardiotoxicity of that molecule, I don't
That
have
will
data
not.
for that. Now if I have to have a good machine learning model to predict these toxicity profile, then I need to have a model which is trained
To
right,
predict.